The last talk of the session will be given by Leonardo Barneschi. Do you need help with the setup, or
sorry?
Do you need help with the setup?
Yeah, I'm just.
I see that the Ignacio is already on his way.
Yeah. I'm just going to share my screen. Yeah. Zoom. Okay. All right.
Perfect. 15 minutes for you.
Thank you. So I'm going to give a little bit of a different talk. It's not about code or theory development. So I guess I shouldn't be really here, but just it's an application in photobiology of QMM calculations that are entirely performed with the Molcas or Open Molcas. So in our group, we have been studying for quite a while a very specific protein family that's called rhodopsin, and it's the very same protein that allows vision in the human, that's responsive to light. The thing is, it's a protein it is widespread in nature, and different building blocks can produce very different functions in different organisms. At some point, it became very relevant to neuroscience because they started to understand that they could eventually use the light to manipulate the nerve impulse. So the milestone in this sense was the discovery of channel rhodopsin 2 by Leonardo, and it is a light-gated ion channel. So the way this works is basically I can find a way to express these proteins in a neuron, and after that I'm going to shine light on the rhodopsin that just has this structure, 7 transmembrane alpha helices that fits really well in the cellular membrane, and it has a photosensitive center that's the chromophore. That's the retinal covalently linked to a lysine side chain. So we're going to express this into the axons of the neurons, and possibly after shining light, something happens, because maybe you're, allowing ions to go to different sides of the membrane, and so you can eventually trigger or silence the nerve impulse. And lately, they started to focus also on how to use this tool to visualize the nerve impulse, because we still don't really know much about how the brain works. And so for this reason, now there is a high demand for rhodopsins, which are fluorescent, and the fluorescence is supposed to be sensitive to the membrane voltage, so that I can actually visualize with confocal microscopy what's happening in these at the single cell level, or eventually at the population level. But for that is really important that we understand how to tune to enhance the fluorescence quantum yield, because the first protein used in this sense was ARC3, and its fluorescence is super dim. It's like 0.01%, and so recently there's been like a race, a lot of big names that went into the game to try to engineer this rhodopsin and make it more efficient in this sense. That's like making it less efficient for its natural biological function. And also Nobel Prizes Francis Arnold and other big laboratories have worked on mutants with enhanced fluorescence, and they came out with a fair amount of proteins. Those are the ones that I'm going to discuss in this presentation, and they're very nice to approach computationally because they feature a nice range of fluorescence quantum yields values. So from now on, I will just refer to these 7 proteins, which are all mutants of the wild type R3 as the arc set. And there is some further comparison between the least fluorescent and the most fluorescent protein, which will be ARC7. So just a quick reminder of a scheme of the response to light in this molecule. What will happen in general that when the retina is in its ground state and it will absorb a photon, there will be from the Frank quantum to a relaxation along a vibrational mode toward basically a planar minimum on the excited state potential energy surface. And the main assumption in this work is that we can like just compare the radiative and non-radiative decay route as an eventual isomerization that has to overcome a barrier along a specific torsional mode to then get to a decay region that's a surface crossing point, or if the barrier is high enough that it can be trapped in the planar minimum and have a excited state that is long enough to release a photon of fluorescence. And so in this particular class of rhodopsin, like the main 2 geometrical modes that we are interested in is the bond length alternation and the torsion, that's the torsion along the C13 and C14 dihedral that drives all trans to 13C isomerization. So because it's very relevant to this work, I'm just adopting from now on a sort of basic valence bond notation, valence bond language, if you will. So the ground state structure is basically a covalent structure where the positive charge is localized on the nitrogen atom, and then what happens in general after the excitation is that the charge is relocated to the beta ion moiety of the retina, and then what happens after relaxation to the fluorescent state is that we still feature basically a large weight of charge transfer configuration with some covalent structure. So what I'm going to show is how with this model we were able to relate the fluorescence quantum yield with the barrier calculated on the isomerization path. And more importantly, we found that there is a second minimum on the excited state potential energy surface that features a twisted dihedral and a structure very similar to that of a conical intersection that's basically very well correlating with the height of the barrier. And in the end, I just discuss briefly what are the molecular level at the molecular what are the factors at the molecular level that are determining this particular shape of the potential energy surface. And this is where the job really started. So we need structures to perform QMM, and just a couple of these structures were available as X-ray crystallography structures. So the other models were just built by comparative modeling because I forgot to say that we are just going to the to a maximum of 7 mutations from the wild type. And I will also discuss very briefly how we calculate these QMM models. Basically, we have a very conservative approach, standardized and automatic to define our QMM model, but basically what we are going to treat at the QM level is just the retinal chromophore, and then the rest of the protein is mostly constrained, and the atoms are frozen, so that we don't really get far from the X-ray structure, and also the side chains of those residues that are close to the retinal are allowed to relax during the QM optimizations. And basically the geometries optimized at the ClassCF level, and in this work the energies have been corrected at the XMS-Caspitidou level. And just to say, it's a protocol that has been extensively benchmarked. And what I want to say is that we are not aiming for the perfect QMMM model to get the perfect vertical excitation energy. We are more interested in capturing the trends in vertical excitation energies, and if we are able to capture some physics of the real models, then that's going to be reflected in a well reproduced trend. And so, as a starting point from any other development, any further study, the first thing we usually do is just assess the vertical excitation energies and see if they compare well to the experimental values. And we have had a pretty nice correlation in this case. We are in good agreement with the experimental data, especially for the vertical excitation energies. A little bit less for the emission of the fluorescent state, but we are talking a very little difference in emission wavelength. And most importantly, then, as I told before, we went and investigated the first excited state potential energy surface by scanning the torsional coordinate that drives the whole trends to 13C isomerization. And indeed, we find that the difference, of course, between the least fluorescent models and the most fluorescent ones is a barrier which is increasing in magnitude with a very good trend. And most importantly, we found another minima, as I told you before, on the excited state potential energy surface that's twisted, and it features diradical electronic structure, so we call it twisted intramolecular diradical, and it's important because we have a very good correlation between the height of the barrier and the relative stability of the planar fluorescent state and the twisted diradical, and it's very reminiscent of the Hammond postulate in organic chemistry. And indeed, also in this case, we were able to measure pretty well against the experimental data. And yes, from now on, this difference in energy between the two minima, that of course has to be the points which are connected by the minimum energy factor. We'll just refer to them as a reaction energy, because it can really be thought as a reaction on the excited state that can be either exothermic or endothermic, and if the products are higher in energy, the transition state will be more resemblant of the product, and I will have a higher barrier. And this is just what I schematized earlier. We checked. We document the electron structure character by just looking at how this charge is distributed down the polyene chain, and after the excitation, the Frank-Conner relaxation to the fluorescent state, we have a really spread charge, which relocalizes along the torsional mode and ends up at this minima that's basically a pure diradical character. And this is we found this twisted diradical that has not been documented in the literature so far to explain the fluorescence mechanism in this class of proteins. Even if I found out recently that that has been sort of postulated by Professor Gararelli at the University of Bologna. But basically this other intermediate has like the same relationship as the fluorescent state and Frank-Conner state with respect to the conical intersection, and just looking at the topography of the conical intersection in the branching plane representation is we can see that it's just intersected by again the bond length alternation relaxation mode, and is there because the conical intersection we found is sloped in this system, and also it's very nice to to see that it has a pure character because it's located close to the conical intersection that's changing the character according to the geometric phase theorem. So since we have this clear representation of the electronic character of basically all these stationary points on the excited state potential energy surface, we thought we wanted to just demonstrate that we could fit our ab initio data with a very simple analytical model, and so basically we fitted our ab initio data with a simple diabatic representation that reflects what I've been telling you so far about the electronic structure of the molecule. So basically, we can represent this data. So I have a movie that's better. We can represent this data as a mixture of two pure states, so two pure diabatic states, ground state, which is as a covalent character at the Frank-Conner, and an excited state that we are just an approximation we assume as a full charge transfer character, and by defining the very simple 2x2 Hamiltonian, we can represent the analytical adiabatic surface, and which I show here that it basically it is able to reproduce very well all the quantities that we are calculated. We have calculated ab initio, so those will be the reaction energy and like the geometry of the this twisted diradical intermediate and the fluorescent state, and all of these with a very trivial analytical fitting of 2x2 Hamiltonian. And so in the end, we wanted to look at what it's really determining at the molecular level the shape of the potential energy surface, and we did that by comparing the two, the most and the least fluorescent model that feature just 7 mutations, 7 mutated positions. And we couldn't say much. To take into account the contribution of the single amino acidic substitution, but in general, we saw that the electrostatic potential projected by the most fluorescent model is basically perfect to stabilize the fluorescent state and destabilize this twisted radical state. So basically, since I've told you that this twisted radical has all of the charge localized at the proton shift base moiety, it's trivial to understand why the most fluorescent model that has a much less strong electrostatic potential in that area is indeed destabilizing what will be the product of this hypothetical reaction. And
I'm sorry to interrupt you now, but you're talking for more than 20 minutes, so you basically used up your time. Could you maybe rather quickly come to the conclusion?
Yeah, sure. So basically, we've shown that the fluorescence enhancement mechanism is determined by the differential stabilization of these two stationary points on the potential energy surface of the first excited state. And we are pretty well reproducing the experimental trend in measured fluorescence quantum yield. And basically, it's the first time that someone proposed this twisted intermediate diradical structure to explain the increased barrier along the isomerization path. And presently, we can just study it at the computational level because it's so close to a conical intersection that's not possible to study spectroscopy. So I thank you all, and I thank my collaborators at the University of Siena, the group of Professor Oligucci, and in particular Emanuele Marsili, Luca De Vico, and my former supervisor, Laura. So thank you for your attention.
Thank you very much for your talk. There is a question?
Yes, I mean, that's probably a question experimentalists would laugh at, but would one be able to detect that diradical molecule with ER, for example? Or
I don't think so, because it's basically very resemblant of a crossing point. So, like, to my knowledge, that they've not been able to characterize them spectroscopically yet. So, because it's an ultra-fast process. So I've never seen even in ultra-fast spectroscopy paper that they could characterize a conical intersection in these models.
All right, let's thank Leonardo and all the speakers of this session.